Hi, welcome back. We all had our sugar shots, caffeine shots, and sunlight shots, so we can go for the next session with short talks. Again, we will follow the same strategy. I will just stand up at about 8 minutes so that you know to wrap up your talk, and then at 10 minutes we will start with some questions. So the first person who will give a talk is Laurah Ondari, and she basically will talk about empowering bioinformatics in Africa through Bioconductor. So welcome, Laurah.
Good morning, everyone. My name is Laurah Ondari, and contrary to other talks that have been given, mine is going to be very non-technical. It's about the work we've been doing together with Maryna in Africa, empowering bioinformatics through Bioconductor and expanding Bioconductor's reach in Africa. So the roadmap of my talk will be why Africa and why now. For the last decades, high-throughput techniques have placed a scientific premium on bioinformatics skills. However, in Africa, bioinformatics skills and expertise are still scarce. And they are scarce because there's lack of infrastructure and access to genomics training. And in the last few decades, Africa has been generating its own data for Africans by Africa in Africa. So with the expansive data that's in the continent, it is important to have bioinformaticians to be able to analyze this data and get solutions that are tailor-made for Africa. If we look at the bioinformatics user base, as the Bioconductor user base, in as much as Bioconductor can boast of a global user base, we can still see that a majority of representation is still in North America, Europe, and Asia. And although there is some representation on the continent of Africa, it's just a mere probably 100 users per site, or in some cases less than that, and that's represented in Kenya, parts of South Africa, and Nigeria in West Africa. And as of 2025, there are about 8 billion people in the world, 1.5 live in Africa, and yet that genomics training is not as represented in the continent. So there's underrepresentation in the Bioconductor user map, and the continent is still underserved. So this is the first gap that does necessitate Bioconductor training in Africa. Oh, sorry. So Bioconductor does have a very rich training ecosystem from about 2,400 R packages that are downloaded approximately 1 million times every year, to helpful functions in vignettes, end-to-end analysis and workflows. There are various books that can be used, workshops and courses. These are the training materials that we would be leveraging to impart this bioinformatics skills and Bioconductor skills in Africa. And this paper by Tan et al. That was published earlier this year in April gives the key resources and best practices for people learning and teaching biological data science within the Bioconductor community. This was a good resource to leverage because we have people who are just starting out to use the Bioconductor training ecosystem. So it does shows like prerequisites, how to analyze data, developing packages, joining the community as learners and also as instructors, which we will see how Bioconductor has come into the continent of Africa in form of instructors for the Bioconductor material. But for teaching, it does give the best practices for leveraging Bioconductor resources to conduct Bioconductor-related training. Oops, sorry. Oh. So in 2022, Bioconductor partnered with the Carpentries to be an official member organization. And from that, it did receive the Chan Zuckerberg grant to be able to get 30 instructors within a period of 2 years. Within that period, 31 instructors were trained globally. And the aim of that partnership and the grant to train instructors was to build local capacity and empower regional educators so that they can be able to expand bioconductors' reach and bioconductors' training materials to other parts of the world. And with that initiative, there were some instructors that were trained within the continent of Africa, some being from Zambia and Nigeria. And with the training of instructors, we were then able to conduct bioconductor trainings on the continent. So the second gap is that even with the partnership between the carpentries and Bioconductor, there was still no Bioconductor carpentry workshops in the continent. You can see some were in North America, Asia, parts of Europe, and even South America. But in the continent, there were no workshops there. So within Bioconductor did get another grant funded by the Chan Zuckerberg Initiative to deliver in-person introductory workshops in East and West Africa. And it should have begun with a focus on introductory workshops and then specialized topics as that partnership went on. So Bioconductor did collaborate with some African-based bioinformatics leaders like my boss here, Trusha, and others like Zedayas and Umar. And this is the paper that shows that funding and how it would be utilized. So we then conducted the first Bioconductor workshop in Africa ever, and this was in Kenya between March 24th and March 28th at the institute where I work at, the International Institute of Tropical Agriculture in Nairobi. This was a week-long course that was aimed at teaching introduction to RN Bioconductor and RNA-seq data analysis. For the first 2 days, this would be the main module taught, and then day 3 and 4, this RNA-seq data analysis would be the module taught, and then on the last day, we encourage participants to come with their own data so that they can apply the material that they were taught on the first 4 days to their own data as a way of seeing if whatever skills that were imparted would be applicable to their data. We had a blend of local and international experts as participants, and to underscore the importance of this workshop, we had 145 applications, from which we had to only select 32 places. So the participants that we had were complete beginners in R, and some had an intermediate understanding of R coding in the R program. But then on day 3, we quickly learned that we had to split the 2 groups because the intermediate learners were learning a bit faster than the beginners, so we had to split this so that the beginners can also have the skills being imparted better without the very fast pace of the course. Our learners did appreciate the learner-centered approach. We had a great deal of good feedback. From that, people were able to say that they could manipulate some R functions. These were people who were complete beginners, and from this workshop, they could be able to code and produce some visualizations in R. And these are just some screen grabs from LinkedIn of people saying how this course was beneficial to them. So as a result of that, Bioconductor has partnered with Physilia, and the main thing here to focus on is that there are 2 free places per Bioconductor course for people from low and middle income countries, which many countries in Africa are a part of. The second thing is that we were able to conduct another Bioconductor workshop in Addis, and this was almost similar with an introduction to RN Bioconductor and then RNA-seq data analysis, and this also had a blend of local and international experts. So the next steps is that we are going to move. We have now had 2 Bioconductor workshops in Kenya and Ethiopia, so we'll be moving to West Africa in Benin November of this year. And because Benin is a majorly French-speaking country, we'll have some translation to French that will be led by Laurah and his team. And in 2026, we look to having advanced workshops and webinars So we are presenting this here because we will be seeking instructors for that. Oops. So we would like to engage all of you. If you'd like, you can join as a trainer or content contributor or collaborator. We are looking for partnerships, and if you can partner with us to address resource gaps or regional needs within Africa, please reach out to us. And thank you. You can scan this for slides. Happy to talk with all of you. And if you want to partner with us, the best person to talk to would be Maria, who invited me to give this talk, but we can have a talk as well. Thank you.
Thank you for this nice talk. Any questions?
So I wish to ask, what were the challenges you faced while organizing the first two sessions and also the forthcoming sessions? And I also wish to know what's the most common background among the participants who applied to take part in the course, in the training workshops?
So I will talk for the Nairobi course where I was part of the instructors. The most common background that we saw are people from biology, molecular biology. We did not have a lot of computer science students, but also that could be a bias that we were holding it at a biological research institute. So that could have skewed the data that we got. The challenges, as we had seen on the slide that I showed you, we had to have some remedial, I don't know if that's a challenge or it's a good thing, but we had to have some remedials for people who are complete beginners because of the pace. But that really helped them to get the skills at par with the people who are already intermediate programmers. Yeah.
Another quick question? No, then, okay. Then we will pass on to the next speaker. Our next speaker is Luca Marconato, and he will talk about advancing spatial biological analysis across programming languages. So the floor is yours. Thank you.
Hello, everyone. I'm really scared about this clicker. I guess I'll just wait a bit. Well, I can go over the title slide, really. So today we'll be talking about interoperability across programming languages for spatialomics, and I will present some work that I did with my colleagues at SCVerse, and also in collaboration with different communities, in particular Bioconductor. Okay. Yes. So in the recent years, the interest towards spatialomics has been increasing at a steady pace. What I show here is the number of publications as from PubMed on the field of spatialomics. And what is also increasing is the number of technologies that are available for profiling this data. This list is for technologies from industry, academia, but is not complete and also is larger by the fact that some technologies come with different versions. On disk, this type of data requires substantially three kind of informations: expression matrices, geometries, and raster data. And on disk, the extensions that are used for this are of different types, such as for raster data, PNG, ZAR, H5, and so on. The complexity increases by the fact that you can take the same sample and profile it with a combination of the technologies that you see here on screen, giving multimodal spatial readouts. And a consequence of all of this is that, I go a bit behind here, is that if you have two technologies and two softwares that are made for each particular technology, more often than not, it's not seamless or easy or even possible to reuse the software across different technologies. And we want to address this challenge, so our goal is really to reduce fragmentation and improve interoperability. To address this, we decided to team up with different consortiums and groups. So as part of SEverse, we team up with OME, Open Microscopy Environment. They have experience with bioimaging. We team up with NAPARI. They have experience with interactive visualization, and recently Bioconductor. So a few words about SEverse. SEverse is a consortium for open source development in the field of single cell and spatial omics, and we developed and maintain a series of highly used Python packages. Something was missing for spatial, so we decided to build a new package that is called spatial data. And the design behind the package is that we abstract the representation for spatial omics into simpler components, reusable components. We call them spatial elements. They can be raster geometries, they can be vector geometries, or tabular annotations for mapping tabular information on top of the spatial information. We can also arrange these elements in space via registration and coordinate transformations. And when we write this on disk, we use some language-agnostic technologies such as ZAR and Parquet, following an open specification, an extension of the OME and GFF specification. A few words about OME and GFF. OME, Open Microscopy Environment, Next Generation File Format. OME and GFF is a specification. OME ZAR, will come later in the talk, is the implementation in ZAR of OME and GFF. The reason why we need this is that if you have a large tissue block and we analyze this with some microscopy, what we want to do is that we want first not to store this in a monolithic file. This would be inconvenient, and also we want to have an organization in space that reflects the user's patterns. We don't want, for instance, to slice this across channels. Otherwise, for looking at a single portion, we need to load the full channel. So what OME ZAR does is that it chunks the data with contiguous blocks that can be accessed in parallel and also conveniently from the cloud. Around spatial data, we built in SEverse an ecosystem that starts from raw data to analysis and visualization. So in particular, we have Spatial Data I/O, which is a package to ingest data from common commercial formats. For analysis, this we do outside the spatial data package with SquidPy, ScanPy, SCDI tools, but also for both data ingestion and analysis, there is a rich ecosystem from the SEverse community. So third-party developers created a lot of ways to analyze this data that is anchored by the SEverse data structures. For visualization, we have two packages, one for static visualization with matplotlib and one for interactive visualization in NAPARI. But what is really exciting is that thanks to the language-agnostic file format, we could explore interoperability across programming languages. So this is a picture from one of the latest hackathons that we did with folks from Bioconductor and other friends. And we explored interoperability in R and also JavaScript. So Elena led an implementation together with the rest of the team for implementing readers and writers of spatial data format in R. I want to stress that this is still more of a prototype implementation, and we actively look for contributors or people that would like to experiment with this. This allowed to use a visualization technique from R, such as ggplot, and then we have VTES in JavaScript to analyze, to visualize this type of data. What is also possible is that we envision the possibility of a full path like the one we did above from raw data, thanks also to interoperability with spatial experiment, spatial feature experiment, and the Voltron, and analysis within R. So then, as an R user, you could reuse some of the readers from Python and vice versa. I want to spend a few more time on visualization. So for visualization, what we have in Python is first a package called spatial data plot, which uses matplotlib. And we have extensive testing for graphical testing. Somehow this misalignment with the legend made it in the latest release. So I thought that it was a good example to really highlight the pain in Python to work with matplotlib. luckily, ggplot is available, so when the full interoperability will be set up, many Python developers will be happy to use ggplot. What we also developed is Napari spatial data. So you can see here we are doing screen sharing in iPad. We can very fastly visualize large microscopy images of aligned layers, and also we can annotate them in a smooth way, and these annotations are all saved together into the spatial data object. And then also what Mark Keller developed in collaboration with SEverse is VTES. So this allows to visualize data from the cloud from a single URL. And it's really powerful because it's not just for spatialomics data, but also you can add analytics on top of it. And Mark and Selena Luo also developed EasyVTest recently. It's a fresh package that makes the configuration of the viewer much faster. I want to spend also some more time on the internals of the R implementation of spatial data. So what we need for reading in R or any language the spatial data disk format is being able to read the various components and then the container object. So on disk, the components are saved in ZAR, Parquet for a vector data, or GeoParquet, which is Parquet with a geospatial extension, and then the tabular information is saved in ZAR with the NData ZAR format. The container is following the OMI ZAR metadata structure, which is basically a set of nested folders with some JSON metadata that are telling what is inside the various container. So to read this, in R, there is the R library from Mike Smith that allows to read the raster data. For Parquet and GeoParquet files, Arrow can be used. And then for tables, there is a combination of single cell experiment, Zell converter, or NData-R and PZAR. Unfortunately, there is no external library for OMI ZAR, so they have to need the parse information from scratch. So a few limitations here is that ZAR currently does not support the latest version, ZAR v3, but so it's on the way, and OMI ZAR support could benefit from a centralized implementation. So what we do in Python is that we have, luckily, a series of libraries to work with OMI ZAR and then internally also ZAR. But there is a problem that OMI ZAR only supports NGFF 0.5, and without being technical, spatial data needs the latest version. So we partially implemented. This partially creates a lot of problem because it's not fully aligned. So what we want to do is not to have this direct link, but put our code in a package that we are building together with the NGFF people to have standardized validation and parsing of the JSON metadata with the OMI ZAR. And the cool part is that not only this will be a cleaner way using Pydantic, so it's a power validation method, but also Bioconductor could leverage the articulate this package, and then this maybe could simplify maybe having a package like this or using it directly. So, conclusion, spatial data promotes interoperability with Bioconductor for data ingestion, analysis, and visualization. Next steps in Python will be a canonical parser for OMI ZAR metadata, which can be reused across languages. And in Bioconductor, ZAR and reusing the Python tooling could improve the developer experience. With this, I want to thank the SEverse team, SEverse community, the bioimaging and GFF community, the Bioconductor team, and all the attendees of the Spatial Data Hackathon. And thank you for your attention.
Thank you for presenting this interesting initiative. Questions?
Hi, thank you for such a good talk. I just wanted to ask, how did you overcome the problem that we heard in the first talk where they had to use 3D dice to make sure that they have the right alignment? What did you do with the spatial transcriptomic data when you had all the files in 2D to make them align in the same format in the 3D?
In the spatial data framework, we have preliminary support for 3D data. So raster data uses, allows to have 3D blocks. But in the initial version, there was not the possibility to have what we call 2.5D data, so have different slices aligned with each other. But we are working on this, so we will present this for the first time next month at the MBL at the ESSB conference. So we've been working on extending this for 2.5D data, and what we do is that we have a link with NeuroGlancer, which is a viewer in the browser from Google that allows to visualize 3D meshes, 3D points, and also have a 2.5D visualize. So it's coming.
A question? No, then I thank you again, and we will switch to the next speaker.
The next speaker is Peiying Cai, and she will talk about differential detection, the detection of differential spatial patterns in spatial omics data. So the floor is yours.
Hello everyone, my name is Peiying, and today I will present the framework we developed to detect differential patterns in spatial omics data. Before I introduce our framework, let me first explain two key concepts: spatial domains and spatially variable genes. In spatial transcriptomics data, it's often possible to label tissue region manually or by clustering cells or spots based on their gene expression and spatial coordinates. And the data set shown here is from human dorsolateral prefrontal cortex. It's generated from 10x Genomics Vision platform, and it spans 6 neuron layers and white matter. And spatial variable genes are those whose expression profiles varies across the tissue. And here are examples of marker genes for gray matter, white matter, and layer 5. So you can see their log transformed expression reveals clear spatial pattern. And our original framework called DSpace is designed to detect such spatial variable genes and key spatial domains affected by spatial variable genes. In other words, we test whether the average gene expression is significantly higher or lower than the average gene expression in the remaining tissue. And here are, for example, spatial variable genes detected by DSpace. Lines highlight the domains being detected. And now we extend this framework because more generally, omics data could often collected across multiple samples and multiple conditions, and it's typically of interest to detect the differences between conditions, and such as genes show different abundances. So we are interested to detect such differential spatial pattern genes, those whose spatial expression patterns change across experiment conditions. Here are two simulated examples for DSP genes. Each row represents a gene, and they say we have two conditions, each have 4 replicates. For the first example, the gene is highly expressed in the inner region under condition 1, and the expression is uniformly distributed under condition 2. And for the second example, the gene is highly expressed in the outer region under condition 1, but shifts to bottom region under condition 2. And those are kind of DSP genes we would like to detect. In the short summary, what does DSpace 2 do? We have 2 tests. First, global test is to detect DSP genes. And the second is individual cluster test. We aim to identify the clusters where gene abundance vary across conditions. Then how does DSpace 2 work? Start from spatial transcriptomics data, where tissue is partitioned into several spatial domains. Then we aggregate those expressions in each domain, producing a pseudobulk abundances. Then we do this step for every sample. And for each gene, XID denotes the pseudobulk abundance for sample I, domain D. We then fit a negative binomial model with 3 sets of covariates for domains, conditions, and their interaction. The domain coefficients model the gene expression spatial structure, and condition coefficients capture the different abundances between conditions, and that their interaction captures how spatial pattern changes across conditions. To detect DSP genes, we test whether the interaction differs from zero via a likelihood ratio test with HR. And to identify the key spatial domain affected, we test whether the corresponding interaction differ from zero for each spatial cluster. Multiple testing is controlled with FDR correction. To evaluate, we built a semi-simulation study using 2 real data sets as anchor data, and then spatially reallocating those real data measurements, simply just swapping cells for each gene. And we created 4 different categories of patterns, 25% of genes in each category. 2 of those categories represent DSP scenario, so where the gene expression patterns differ between conditions, and the other 2 are non-scenarios where the spatial patterns are same across conditions. It could be that in both conditions, the expression is uniformly distributed, or they have the same highly expressed region. After generating such simulated data, we re-estimate the spatial patterns using clustering algorithms and then use it as input to our methods. Since no existing methods was designed to such DSP detection, so we compared our methods with common marker gene methods. So red fine markers, green fine markers, pseudobulk version of fine markers, and spatially package which use pseudobulk and do differential expression analysis with lemma. And in both cases, here are true positive rate versus false discovery rate curves. In both anchor data and the 2 tests, global tests and individual cluster tests, these based to have good FDR control and show high true positive rate. Beyond simulation, we applied all methods to real data. It's with 5 post-injury time points, and because not every method could be applied to such multiple conditions, so we do a 2-condition comparison, also 5-condition comparison. Unlike simulations, real data set doesn't have ground truth. So to overcome this, we collect potentially relevant genes. In this case, it's healing and regeneration. And then for each method, we use top 200 genes. Then we count how many of those overlap with the potential interest genes. And we found that these based to in both 2-condition comparison and 5-condition comparisons detect more genes in top 200. We also investigated the computational efficiency. As expected, pseudobulk-based methods run much faster. And for instance, these based to runs in 40 seconds to 38 seconds with 6 samples, each containing around 15,000 genes and 10,000 cells. Our differential spatial pattern framework is available as a Bioconductor R package, called DSpace from version 2, and our original work was published in Bioinformatics last year. The differential spatial pattern extension is available as a preprint, and if you are interested, you could scan the QR code, reach out our vignettes, and preprint. At the end, I would like to thank Mark and Simona and all members of the Robinson Lab. Thank you for your attention.
Thank you for this great talk. Questions?
Thank you very much for the the presentation. I'm sorry if I missed that, but the whole analysis seems to strongly depend on how you define the spatial domains in the first place. Is this part of the algorithm, or do the user have to provide these domains?
Yeah, so it would be the input for the algorithm. So like we highly based on those spatial domains as input, yes. And I think that usually is the case because there are many clustering methods to detect spatial domains, and also often you would have manual annotation. Yeah.
There was a question in the back.
I was going to actually ask something really similar. Do you have a personal preference on how spatial domains have to be trained as an input before running this? You know, some people actually would build a really, basic neighborhood around cells or spots and just count the identities and build that as a, niche kind of profile to do the spatial domain clustering. Mm-hmm. But Banksy, I think, I just checked the manuscript to see which kind of examples that you provide. It could be as input to spatial domains. Do you have a personal preference on basically what would be the best spatial domain to put as an input for this?
I think based on like prior knowledge, because here we use multiple samples and multiple conditions, so you would have like require the reasonable domains that as the input. So I think if the cluster is very messy, then maybe that's a problem, right? And if it's like as clear and smooth as possible, I think that would be fine. Yeah.
Other questions? Then I also have a question. I was wondering if you have seen problems with respect to double dipping. So if you first do the clustering and then the differential analysis, do you expect problems there as well?
We do like non-distribution tests, and we don't see these kind of issues in our test. But yeah, that's.
And would that be because you test for the interaction mainly?
Yes, that could be the one possible reason for it.
Okay, thank you very much for this interesting talk. And maybe let's give a hand, and then we switch to the next.
The next speaker is Martin Emons, and he will talk about differential spatial co-localization analyses.
Okay, hi, I'm Martin. I'm a PhD student as well from Zurich, from the group of Mark Robinson. And I will talk today about differential spatial co-localization and the package that we wrote to achieve this, which is called spatialFDA. So I present you a small TLDR. So spatialFDA is a bioconductor package to assess spatial co-localization of cells. The methodology combines point pattern analysis and functional data analysis. I will show you simulations that we did in order to understand the performance of spatialFDA and its competitors. And if time allows, I will briefly show a biological use case of type 1 diabetes and looking at the co-localization of immune cells and pancreatic islet cells. So I say here that we are using point pattern analysis, so then the first question is, well, how do we get to points? And together with Samuel Gunz, we recently published a paper lining out a little bit how we think about spatial omics data. And for point pattern analysis, we think of it in two ways. So you can either approximate the features directly, so you take the XY coordinates of your transcripts, you give them an identity, which is the transcript identity, and that's how you build a point pattern. This is a very reasonable thing to do because transcripts do not really have a size or a mass, and you can approximate them well as a point in space. The other avenue is to collapse the cell into its centroid and to assign the cell type as the categorical variable of your point pattern. This approximation, I think, is reasonable for roundish cell types with uniform size that do not vary. Maybe you would not want to approximate your neurons with the centroid. So that's one caveat of our methodology, the approximation is points. So then, with these point patterns, yes, we built this framework, spatialFDA, and the basic idea is we have several samples, so you could have different images. Most often we might have several images per patient, per sample. And then what we do on these point patterns is we calculate these spatial statistics functions. These spatial statistics functions are usually on the x, they have a radius, and then on the y, some edge-corrected estimate. And then, as I said, this is for a multi-sample framework. We calculate one curve per image. And because you might have several images per patient, we have this setting that several curves actually correspond to one patient. So these are non-IID samples. Then the question is, how do we compare these curves? And I guess a natural way to do that would be to collapse the curve into a scalar value and compare the scalar value with, say, a linear model. We decided to do it a bit differently, and we used functional data analysis to do that. So functional data analysis is basically the idea you do not compare individual scalar values between conditions, but you actually compare the entire curve or the function over its domain. So as well, the output of a functional data analysis model, the coefficients are themselves functions. So we implemented functional PCA, which basically extracts the main modes of variation in your functional data, and you can get a rough clustering of, well, which curves are more similar to each other and which are not. The actual inference we perform with a functional generalized additive mixed model, where you estimate an intercept curve, so that's the kind of generalization to functions, right? And then the coefficient is as well estimated as a functional effect over a domain. And the kind of nice advantage that you might get out of this is that you understand the scale of your effect. So here you might see that in comparison to the control, the perturbation has an effect at larger radii only. So we wanted to understand a bit the behavior of spatialFDA and of competitors, and in order to do that, we created a multi-sample simulation. And I do not want to go into too much detail, but the simulation is based on three kind of pillars. So the first one is random circle packing, where we pack random circles in space to account for cellular spacing. The second part, what we added on top, is a multi-sample parameterization. And basically what we can do is we can create different conditions. So here we might have a control where cells here in red are interacting with a probability of 0.9. And then we can have null simulations where the actually interaction probability is similar with some sampling. And then we create differential effects, so increasing perturbations until at one point we have only a 10% probability of interaction. Indicated in the rectangles is that we can have some variability, and long story short, what we end up is we have conditions with multiple sections and multiple samples to account for some variability between conditions, and we have that over increasing condition effect. So what we do with this is we compare this then to other methods and to our method. And the first thing that I included is a baseline. And the baseline is quantifying the average intensity in the picture. So average intensity being the number of points given the size of the image. And then I compare them with a t-test. This was the most basic way of comparison between these spatial point patterns that I could think of, because intensity is somehow not quantifying cellular co-localization, but it might hint at two things. A, can our simulation capture differential co-localization effects that are not just differences in intensity? And B, are differential co-localization methods more sensitive than a very basic t-test? Then the two other classes of methods are those that quantify the curves into a scalar value and then compare them with a form of a linear model. And the second class is those that actually take the entire curve and then compare them with a functional model. And that's where the two versions of our method, spatialFDA based on an L function and spatialFDA based on a G functions come in. So, yes. So with these perturbations, I create power curves. So let's look at one power curve. We have here the difference in mean probability. So 0 means a null simulation, and then you have an increasing difference effect. And the y is the power. These three facets are the number of cell types. So the proportion here, the cell type that I perturb is rather rare. It's only in 10% is this cell type, then 20 and 40%. And when we look at the blue curve, that's actually the intensity t-test. In the low proportion scenario, the intensity t-test doesn't pick up any differences, which is good because our co-localization metrics actually do pick up a difference that hints us towards that our method can actually simulate differential co-localization without just changing intensity. When we increase this proportion, you see that this blue line actually becomes the most sensitive method, so that basically in the high proportion, cell type proportion scenarios, what we simulate is a difference in intensity, and that's what's happening. Irrespective of that, in pink you have spatialFDA based on a G curve, and it is behaving quite stable over all these scenarios, and I would claim that even in this case here, where the intensity t-test is a bit better, spatialFDA with a G curve is on par or comparable. If this jumps, okay. Very briefly, I just want to show you what we can do with this in terms of with biology. So we have here a data set of type 1 diabetes with 3 conditions, non-diabetic, onset, and long duration. And we run spatialFDA on all pairwise combinations. And what we get is these kinds of heat maps where the color is the co-localization effect. Red means more co-localization, blue means less, and the size is the p-value. And what we just notice here is we have the pancreatic cells here and the immune cells here, and we see that there is an increased co-localization of immune cells to pancreatic cells in onset diabetes, which is absent in long-duration diabetes. I told you about the scale effect. I will jump over this, but we get for each of these comparisons then as well the scale effect, and we see that this difference actually is at lower radii compared to high radii in onset diabetes. So yes, we have done this. We see that we have this immune cell recruitment in onset diabetes, which is less frequently shown in long-duration diabetes, and spatialFDA offers this scale effect, so that these differences are actually significant at radii smaller than 100 micrometers. And this is kind of a nice validation because this is in line with the original paper, Damon et al., which postulate this immune recruitment. Yes, so we've built this. It's an R package. It shows an overall good performance. And due to, whoops, nope, not backup slides. With that, I would like to thank everybody involved in this project, the group from Mark, Samuel, who helped me a lot, Isa Schoon, and as well Fabian Scheipel from Munich, who has been very important in the functional data analysis part of my project. Yes, thank you.
Questions? Yeah.
Thank you. So you view these images as point patterns, and then you create features from these. Could you expand a bit on the 2 features you mentioned? So you saw the radius, and you mentioned edge correction, edge corrected.
Yes.
What do these features mean, and why are they important to assess differential co-localization of these cell types?
So I glossed over that part maybe a little bit. So maybe you're familiar with Ripley's K function. It's a way that you take a point and you just draw a radius around it and you count how many cells fall in that radius. So that's just the raw count. And you do that for all points and you get an average estimate of how many cells fall into it. You increase the radius and that's how basically this curve is constructed. Now, at the borders of an image, you would expect less neighbors on average. That's why you have edge corrections. So that could be isotropic correction or border correction, where you just account for these edge effects. That's what I mean. So it's basically the raw counts, but corrected for border effects.
Okay, so the next, give a hand again. The next speaker is Ilaria Billato and she will talk about the standardized Bioconductor framework for integrative analysis of histopathological images with multi-omics data. So the floor is yours.
Hi. Okay. So hi everyone. Thanks for the introduction. And today I talk about this project. It's a new one. We started working last October. And we work with images. So digital pathology is very important because it gives us the opportunity to analyze images in a different way and give us a lot of information that can integrate with omics data. There are several types of images. We have H&D images, TC scan, or immunohistochemistry. But for this part of the project, we decided to focus on H&D images. We create a Bioconductor working group. It's a multi-omics integration of histopathology image analysis. We have a channel on Zulip. If you want to join us, we share some information sometime, and we have a twice monthly meeting. And yeah, that's going on. So first, what we decide to do, we have to decide which data use as input data, and we decide to use the TCGA images. Why we use TCGA? Because TCGA gives the chance to share a lot of data, like omics data, and there are 11,765 diagnostic images in which there are 32 different tumor types, and they're saved in SVS format. There are other packages in Bioconductor, like TCGA Biolinks or curated TCGA, that give you the chance to integrate these omics data, like methylation, bulk, copy number, in your R session. To download all these images, we took like 25 days, and they occupy like 11 terabytes, so you need a lot of storage to deal with these images. And okay, so these images, we use the 40x objective. Then when they take the sample, you have the top and the bottom section that are processed like a 20x objective, so they are low one resolution. But we take the one in the middle, that is diagnostic FEFPE, that is the 40x, so the high quality. We decided to use the high quality because, yeah, so we can have more accurate result. Then we decide to extract some features. So from these images, we need numbers to do some analysis, and we extract features like human readable first. Human readable are just numbers that has a biological meaning, as the name said. And we first use HoverNet for the segmentation. HoverNet is a deep learning model, and it gives us the chance to make the nucleic classification. So we have benign, epithelial, neoplastic, stromal, and necrotic. As an input, we give the whole slide images, and the output is a JSON file. In the JSON file, there are the coordinates of each nuclei, and yeah, and the annotation. So that's the result. So this one is the input images, and this is the result from HoverNet. This one is just coordinates of the nuclei and colored by the annotation like benign, necrotic, no label, inflammatory, neoplastic, and stroma. Then we use this JSON file as an input in Python with SquidPy, the scikit-mate, to extract some biological feature, like a spatial feature, like nearest neighbors, distant to closer nucleus, and intensity feature like mean, variance, intensity. Then we want to use also QPath. QPath is a very user-friendly platform in which you can do your segmentation, extract some features. And yeah, the input, it was a JSON file that we have to convert in a GeoJSON file, and the output is a GeoJSON. Then we want also to extract some embeddings. Embeddings are number more or less close to the t-SNE or UMAP in single cell analysis. And yeah, you can have embeddings at slide level or tile level embeddings. So you can divide the images in very little parts. I have embeddings for this small square. Then, okay, the input is a whole slide images, and the output for now is a SVS file, but could be a parquet file or whatever. We should find the best way to store these images and result. And then we want to integrate everything in image TCGA. Image TCGA is a Shiny package, is a Bioconductor package now since April. And yeah, there is a Shiny application, and you can explore the data. From here, if you scan the QR code, or here is the link, you can have access to the Shiny app. You can filter here the project. For example, if you want to work with ovarian cancer, breast cancer, or whatever, you can select here. You can select the sample type, the source site, and yeah, and this is the output. And you have the total records, the number of samples, the number of images. And then if you click here, you can visualize the images from the TCGA that, yeah, just it's a link to their website. Then we want to focus on a case study, so we decided to focus on the ovarian cancer. For ovarian cancer, there are 107 images, and there are also available the RNA-seq data. And also there are a lot of information that we can integrate with this data. For example, the image feature, the clinical data from the TCGA, the survival data. We can use consensus ovarian cancer that give you an annotation on your sample, genomic supersignature, and integrate with the copy number alteration signature. Just a first check, we compute the purity overnet is just the number of neoplastic nuclei on the total number of nuclei, and we call that purity from overnet, and we correlate this purity with the purity from absolute, lump, and estimate. Lump, absolute, and estimate are some purity metrics based on, for example, copy number data, methylation, and transcriptomic data. And the correlation is not so high, but in this particular case, the p-value is significant, so it's lower than 0.05, so it doesn't mean that there is a trend, so the result that we obtain with overnet has a biological meaning. Nope, nope, nope. Okay. So for the time to download, to do all this stuff, for the download we took like 1 month. For the overnet, 6 months. For gigapath, 6 months. For the SquidPy extraction, like 3 months. To convert GeoJSON, GeoJSON, like other 3 months. For QPath, we estimate like 7 months. And it's a total of 26 months. So if you want to do all this stuff, you take like 2 years. But if not, you can just download the package, and you should have access to all this data. And for now, oh God, okay. That's funny. Okay. Okay, so more or less we are done with all this stuff, and just we have to wait for QPath. And then, yeah, there are other repositories for images like the TCAI data. They are another archive. And if you want to know how to download, how much time it takes to download all that images, you can use TCAI API. It's another package. It's on GitHub for now, and we are thinking to submit maybe for the next release, but if not, the next one. And you can use this package to download all the SVS images from the TCAI archive. And so, just in conclusion, yeah, the features are more or less soon available on Image TCGA. If you need already them, you just text me or one of my collaborators, and we can share them. And also, we want to integrate new function in TCAI API, just also to visualize the image. And yeah, more or less that's it. Thanks.
Thank you for introducing this great data resource. Any questions?
Thank you, that's interesting. So my question is more technical. If you can tell us, how did you kind of set the number of layers and the bottleneck in your model, given the structure of in your deep learning model, given the structure of the data?
Okay, so they are just public model, so we didn't change anything on that model. We just use and take them for the analysis on H&D images. So, yeah. So if you want to set other things, you can just explore your model, try to change the number of layers, but yeah, maybe I don't understand, so yeah.
Okay, we can talk later about that.
Yeah,
Thanks.
okay, that's okay. Yeah, no, but actually, I think that these models are very helpful, but they require a lot of memory usage, GPU, so you have to reach a lot of cluster, a lot of resources, and they takes a lot of time, so.
Yeah, sometimes adjusting the number of layers between the input and the output could also have significant effect on that.
Yeah, yeah, exactly.
So that, okay, fine.
There is another question here in the front. So.
Thanks a lot. Do you know by chance if bulk RNA-seq data is generated from the same tissue that you have the slide for?
Yeah, it's generated from the same sample, yeah. If not, they change the codification of the sample. So at some point when we want to create this case study, we merge all the data. So in that case, we not work anymore with 107 images, with just 82 images, because the merge, the corresponding between sample and images is 82. So, yeah. But you can check that, yeah.
Thanks a lot.
Question there in the back.
Oh, thank you for the presentation. So you use the HoverNet for supervised fashion. Yeah. And you use for gigapet in the unsupervised fashion. But ultimately, you should be able to generate embeddings, even though you don't do any classification of sorts in the HoverNet. Can you extract those embeddings, or let's say that you can compare them with the embeddings that gigapet generates?
Yeah, I think that if you want, you can also extract some embeddings from HoverNet. But for now, we just decide to use the classification from HoverNet. But yeah, I think you can do.
Okay, so give Ilaria another hand. And our next speaker is Maryna Chepeleva, and she will talk about functional signal decomposition in spatial transcriptomics and multi-omics data. So the floor is yours.
Thank you. Hello, everyone. I hope it will work. So my name is Maryna. I'm doing PhD in Luxembourg Institute of Health in Multi-Omics research group in Department of Cancer. So cancer is our main research field. And yes, one of the key challenges that we all face, especially in cancer research, is that our data is highly heterogeneous. So tissue is not uniform. It contains of diverse cell structures, different cell types, and even and each cell have their distinct molecular programs. And even if we go in for single cell analysis, we know that inside cells there are thousands, hundreds of pathways which overlays and hidden in each other. And standard omics measure average of these signals together. So the question is how we can identify these underlying biological processes from the mixed data. And one approach which could be useful here is unsupervised data-driven approaches, which allows us to deconvolute complex signals. And in my lab, we provide a tool which could be used for this. It's based on independent component analysis. Briefly to remind you how it works. Imagine you have mixture of several signals. It could be voices of the people. Then you can record them on several microphones. And then independent component analysis allow us to separate the signals. It extracts as independent signals as possible. And we will have the voices of these signals that we have initially. And this approach works pretty good with biological data. It could be different omics. Yes, sorry. So speaking about the matrices, we have our gene matrix, proteomics, metabolomics, whatever, with features and samples, and then ACA decomposes matrices into two. First matrix contains independent components in columns, And these components could be linked by functional annotation with different biological processes, cell types, clinical data. And second matrices is a weight matrix of each sample in our new dimensions of independent components, which could be used for machine learning algorithms to classify patients, to predict survival, also could be combined with embeddings from images, whatever, for downstream analysis. And we did this ACA consensus, which means robust, because we ran it inside many times to provide as robust decomposition as possible. And this method was packed into Consica, our package. It's available in Bioconductor. You can load the different data modalities, also bulk and single cell, compare data set. And what we do as extension, we provide automatic reporting for each component after your ACA run, so you can quickly see did your signals which you extracted have some biological meaning, and also linking with your clinical data and metadata. And we have a paper with some examples of successful examples of the usage, and today I will show you some new applications that we tested from last year. And for integration of data modalities, if you have several data modalities, we propose a late integration approach where you run ACA separately, and then depending on how you can link your these data matrices, we can do similarity analysis via correlation, for example, also link clinical data from one of the data set, and also we can combine weight matrix with one more modality and search for some hidden linkage which we don't see before deconvolution. So we applied this method for spatial transcriptomics. For this pilot study, we took some data slides from Hist1K dataset. The idea was so we have a whole slide images with a lot of spots, which contains transcriptomics. And we just ran Consica for this whole slide, and each spot was considered as a sample without any information about dimensions like X and Y. So this is one example. We had this whole slide image of melanoma. So for me, I don't have idea what here. When I just look in it, I need a pathologist. And one of the components that we find here was component linked with currency differentiation. And we see that it's shown us exactly where we have a epithelium layer. And also if you're looking for pathology, we see that this is where we call our epidermal layer is co-localized. So we can define some tissue architecture. Another component which is also very interesting is component which corresponds to immune response, and we have these highlighted areas with red, which show us that this is high areas with high immune response. And again, if we go in for image, this is areas with high lymphocyte infiltration. So now we can see that this is the tumor part, and so lymphocytes are around it. Also to check if our spatial organization not random, we calculated more on index, which show us that we have significant spatial organization of our component. And most of all components, we have it high. Then we did comparison with another method. We took GCA, we did single spot GCA for the same slide. Took for illustration adaptive immune response. We see that it's more diffused region, and comparing with Consica provides us more precise and high resolution of these regions which have a high immune response. And I would like to show you one more example of multi-omics linkage. We took data set from our collaborators. This is glioblastoma data set with not blood, this yellow from the blood plasma samples from patients before diagnosis. And we had here proteomics data with several platforms and metabolomics data. We applied deconvolution with Consica for our proteomics data, and from S matrices we do functional annotation. And then we combine this functional annotation with metabolomics data and looking for high correlated metabolites with these signals to find some functional links. So from proteomics, we find a signal. I will show you only one just to illustrate how it works. So we find components which connected with pro-inflammatory signal because we find some biological processes with immune cell recruitment, like neutrophil chemotaxis or chemokine signaling, and also some processes for cell growth and proliferation. And looking for metabolites which will be linked with them, we found several of them which are responsible or involved in signal cell migration and inflammation, and also fluid for proliferation and energy processes in the cells. So finally, this component, which was found from proteomics, was very good linked with, positively correlated with metabolites that correspond to each other. So now I'm moving to my conclusions. Consica effectively deconvolve complex signals from transcriptomic data. It can identify both structural tissue signals and also functional annotations like immune response, and provide higher spatial resolution compared to GCA case. Also, it's a tool for multi-omics integration. I show how we can combine proteomics and metabolomics with coordinated signatures from different molecular layers. We identify inflammatory program in our data. And Consica is a tool which could help you to generate interpretable biological hypotheses from complex data set. And our next focus that we are looking for is application on multimodal spatial data set, and we're looking for data set to test it more broadly. I would like to say thank you for my team, for all collaborators, and also for Bioconductor for this conference and the opportunity to present. Thank you so much, and now time for questions.
Questions? Maybe I will open the floor then. I was wondering how well you can interpret the components based on the features that are driving them.
Yes, so as a data-driven method, unsupervised, we never know what we will find. And after extracting components, we run functional annotation, for example, with TopGeo with biological processes, and then you need to scroll through the result. And in our package, we implemented automatically reporting for this to do it very quickly. And also when you do ACA, you usually try a different number of components. So you never know what you will get, but with our tool, you can do it quickly and find something.
And can you also get an idea on the features that are driving the component?
Driving.
I mean, like, can you see the importance of features in the component?
Yes, of course. In S matrices, you have like coefficient for each gene. And these independent components, if you do it for transcriptomics, it will be gene sets with a limited number of genes and with coefficient for each gene.
Other questions? If not, then let's thank Maryna again. And we will switch to our last speaker of this session, to Liyang Fei, who will talk about barbieQ and our software package for analyzing barcode count data from clonal tracking experiments.
Hi everyone. How nice, it's the last short talk today. I'm Liyang. I'm from Peter MacCallum Cancer Centre from Australia. I'm a PhD student supervised by Professor Alicia Oshlag and Dr. Jovana Maximovic. And today I will show you our package, barbieQ, that we developed for analyzing barcode data from clonal tracking experiments. So first I will show you some background. So what's a clone? A clone is a cell and is an initial progenitor cell and all of its progeny cells. Here, one color represents one clone. And the cells, once the clones are treated, the composition of clones can change. And if we visualize this perturbation through via a Sankey plot, we will observe two things. First, some clones disappear, like the green clone, but the others still exist after treatment. And so this refers to the change in clonal presence and absence. And the second thing we observe here is some clones, they don't just exist, they also expand at different levels. And this refers to the change in clonal abundance. And understanding the clonal change before and after treatment reveals some important features, such as drug-resistant tumor clones that persist after treatment in cancer research, or multipotent stem cells in stem cell development research, that these clones exist in under various conditions. So with the need to track the clones, this can be achieved by genetic labeling of clones via semi-random DNA barcodes. The DNA barcodes, they are a synthetic short DNA sequence, and the barcodes are unique, and they can work as clonal identifiers because they're unique, and they can be initially integrated into a cell's genome. And once the cells divide, they get passed to their daughter cells during cell division. So in this way, cells coming from the same clonal family will share the same genetic barcode in their genome. And given that, once we want to quantify the clonal abundance, we can just sequence the barcode DNA. And here, once a clone expands, the barcode expands as well. And counting cells in a clone means counting that specific barcode. And in a typical barcoding protocol, DNA barcodes are extracted and amplified by PCI and sequenced by next generation sequencing. And eventually we get a barcode count table from which a sample is a collection of the progeny cells. And we have barcodes in rows, and the count of barcodes means the number of cells in that sample. So this is a bulk DNA sequencing, it's not single cell. And in a real barcoding experiment, people usually have duplicated sets of initial clones that gets injected into different animal models under different conditions. So these duplicated sets of clones, once they're undergoing different conditions, they may have different clonal output. So in a barcode count matrix, we would observe differential barcode output in the samples under different conditions. And typically we would, it's common to have hundreds of samples in columns and thousands of unique barcodes in rows for a typical barcode count table. And the barbieQ package focuses on analyzing this type of barcode count matrix. So, and the core function of barbieQ is to test the barcode change between sample groups. It's primarily implemented with two tests. One is differential proportion for barcodes, and the second one is differential occurrence for barcodes. So differential proportion basically answers the question that which barcodes show significant difference in proportion in different samples. And differential occurrence basically answers the questions that which barcodes are more likely to be detected under some samples between sample groups. And the differential proportion converts barcode count into barcode proportion per sample, and differential occurrence takes barcode counts as a binary value of presence or absence. That these two tests basically answer two different questions. Yeah, and the two tests, both of them model barcode variability sourcing from multiple factors, because in the clonal tracking field, people usually have quite complex experimental setups. Like a sample could be collected from, samples can be collected from different time points and under different treatments with different levels of replicates. So the two tests use linear regression, generalized linear regression, to account for the variabilities sourcing from different factors. And specifically, differential proportion is based on lemma, and we recommend arcsine square root transformation for the proportional data as it holds the false positive rate better in the barcode scenarios compared with other transformation methods. And for differential occurrence, it's technically based on logistic regression to analyze the differential barcode presence or absence. And apart from the statistical testing, barbieQ also has pre-processing methods and visualizations. So basically, the package takes in a barcode count matrix and saves it in a barbieQ object, which is built upon a summarized experiment object. And based on this object, we can apply a bunch of pre-processing methods, and we can test barcodes, and we can achieve visualizations. So, yeah, I'll quickly show you an example of applying barbieQ to the package data, which is a monkey hematopoietic stem cell experiment, and that's monkey blood stem cells. So you have a bunch of barcoded starting clones, and you inject them into monkey and collect blood from at different time points, and from each blood draw, cells were sorted into different cell types. So one sample is a specific cell type collected at a specific time point. So for example, this data set has 16,000 around 16,000 barcodes with 60 samples, and it has assays like the raw count, proportion, occurrence, and it has raw data to save the barcode features, and column data to save the sample metadata and sample features. It's a public data set downloaded from this paper. And this is an example of barbieQ filtering out low count barcodes. It's a data-driven strategy, and you can see before and after filtering, we have a much better visualization of the important barcodes. And collectively, this method retains 8% of barcodes, which contribute to 93% of the total count. So we didn't lose much information. And this is an example of testing barcodes, for their differential proportion or differential occurrence between the cell types, and taking all the time points into consideration. And this is a collection of the visualizations in barbieQ. Basically, every step is associated with visualizations, and we have additional ways to look at sample correlation and sample similarities. And the package is available on Bioconductor, and we have just submitted a manuscript to bioRxiv last week. And I would like to finish up by acknowledging our lab and colleagues and friends and the organizing committee for this awesome conference. Thanks.
Questions for Liyang?
Great technique. I just, maybe I didn't catch that right. Did you label one hematopoietic stem cell that you injected, or did you label all hematopoietic stem cells circulating in the blood? I missed that. And did you think about studying like aplastic anemia or MDS, where it's actually really interesting to look at the clonal changes in the blood?
For this experiment, did they label every single hematopoietic stem cells? Yes, they did. So, one cell or many? Lots of cells.
Other questions?
I was just wondering, with respect to time, do you often see like non-linear patterns over time? So you were able to include time as a linear covariate in the model, and I was I was wondering if you often see like non-linear patterns of proportions over time, or if that can also be addressed.
Sorry, pardon me.
Okay.
Did I select barcode show significant difference?
You will model the barcodes over time because you have like different measurements at every time step. And I was wondering if you often also see like non-linear changes in the proportions or in the detection over time.
Yes, it's common to see non-linear changes.
And you can also address that with the model currently?
After we transform the proportions, they look more linear.
Okay. Yep. Okay. Other questions? Otherwise, I suggest to give Liyang a hand again. I would like to thank all speakers for